Assalamu alaikum dear student. Today we are going to discuss glycolysis. This is an introduction of glycolysis chapter, and then we are going to discuss the glycolysis pathway in subsequent lecture. So, how we can obtain glucose from our diet? First we are going to know what is the basic, what is actually the glucose is. So the glucose is a simple sugar with a molecular formula of C6H12O6, and it is the most abundant monosaccharide, and which is a subcategory of carbohydrate. So we can say our body mainly consumes monosaccharide. We can obtain the glucose through protein, carbohydrate and fat, which ultimately converted into the glucose, and then the glucose is further processed into the ATP. ATP is an energy currency of the cell. It is required for our proper functioning of the cell. Introduction of glycolysis. It is a metabolic pathway, as we know. Then, an ancient pathway employed by the host of an organism. The of reaction is that one molecule of glucose is when metabolizes, it is converted into two molecules of pyruvate with a net production of two molecules of ATP. This process occurs in anaerobic condition. What do you mean by anaerobic? It means in the absence of oxygen. So, in anaerobic process, this pyruvate is further fermented and it is converted into two parts, that is the lactate through the lactic acid fermentation or lactate fermentation, and ethanol through ethanol fermentation under aerobic condition. Aerobic means in the presence of oxygen. This pyruvate is completely oxidized to carbon dioxide and generating more ATP. Please note down a point. When glucose synthesized from non-carbohydrate precursor, that is through lactic acid and pyruvate, it is known as the gluconeogenesis. What do you mean by gluconeogenesis? Gluco means glucose, neo means new, genesis means formation of a new molecule, right? So gluconeogenesis is formation of new glucose molecule other than the carbohydrate, which means the non-carbohydrate precursor. The glycolysis and gluconeogenesis have some of the same common enzyme. That is, the two pathways are not the reverse of each other. It means if glycolysis is going to take place, then gluconeogenesis will not going to take place. And if the gluconeogenesis occur, then glycolysis won't occur. It is a highly exergonic. Exergonic means it releases lots and amount of energy, and it is irreversible step of the glycolysis is by passed in gluconeogenesis. Please note down this point that it is irreversible step of the glycolysis is bypassed in gluconeogenesis. Both pathways is strictly controlled by intracellular and extracellular signal. Okay? Are reciprocally regulated so that the glycolysis and gluconeogenesis do not take place simultaneously in the same cell at a significant time. The history of glycolysis. A key discovery was made by Hans Buncher and Edward Bruchner, and it is quite an accident. The Buncher was interested in manufacturing cell of the yeast extract for possible therapeutic use. So basically he was interested in extracting the yeast, but he wanted to preserve it without the use of antiseptic phenol. So they decided to try sucrose, which is mainly found in the kitchen chemistry. So the result obtained is the sucrose, which is found in the kitchen chemistry, is fermented into alcohol by the yeast juice. Then the Buncher demonstrated for the first time that fermentation could take place outside the living cell. This was the great discovery. The accepted views of their day asserted by Louis Pasteur in 1860 was that the fermentation is inextricably tied to the living cell. The chance of discovery of the Buncher opened the door of modern chemistry, that is the metabolism become the biochemistry. They want to preserve this, okay, but due course of an accident, they discovered the fermentation. So from modern chemistry to metabolism become chemistry. The term glycolysis is derived from Greek word glycan, which means sweet, and lysis means dissolution. Glycolysis is also known as the Embeden-Meyerhof pathway. This is an athlete, okay? He is running and he required a lots of amount of energy. So while running, all the cell in a body want more energy. So the glucose is converted into 2-pyruvate so that this pyruvate can further transport it to the mitochondria for the production of ATP. Glucose is an important and common fuel. In mammal, the glucose is the only fuel that the brain uses under non-starvation condition. In starvation condition, what the brain is going to use? It will going to use the ketone body. The only fuel that the red blood cell can use at all. There's a question in the mind that Almighty God has created lots of carbohydrate, but why we only use the glucose? Because instead of glucose we can use some other sort of monosaccharide as a prominent fuel. So why it is so? The reason is that the glucose is one of the monosaccharide formed from the formaldehyde under prebiotic condition. So it may have been available as a fuel source for primitive biochemical reaction. Glucose has a low tendency relative to the other monosaccharide to form non-enzymatically glycosylate protein. In their open chain carbonyl form, monosaccharide can react with the amino group of protein to form Schiff base. It is nothing but these are the compounds which are being used as a ligand to form coordination complexes with the metal ion, which rearrange to form more stable amino-ketone linkage. Such non-specifically modified protein often do not function effectively. Glucose has a strong tendency to exist in a ring formation. I will let you know what is the ring formation, what is the cyclic formation, what is an open structure of glucose. Consequently, relatively little tendency to modify the protein. All hydroxyl group in the ring conformation, remember this point, of beta glucose are equatorial, contributing to this high relative stability. How do these glucose molecules make their way into the cell of our body? Our sugar diet consists of polysaccharide from the plants in the form of starch and the polysaccharide from the animal in the form of glycogen. Means we used to eat the vegetables, from them we get the starch. And when we use the chicken, fishes and beef, we are going to get the glycogen. These polysaccharides are too large for the blood transporter and the cell to uptake. Therefore, our digestive system utilizes proteases to break them down into smaller components. Enzymes that are required to break down the polysaccharide into smaller components. One of them is alpha amylase. It is a form of amylase in human and other mammals that cut the alpha bond of a large sugar molecule. These are the two types of starches, that is the branched and the unbranched. The unbranched starch is known as amylose and the branched form is known as amylopectin. Both of them contain 1,4 beta-glycosidic branch and the branching also contain beta-1,6 branching. And when alpha amylose act on it, it is converted into glucose. In animal, the alpha amylase are the major digestive enzyme. It includes two digestive enzymes, the ptyalin. In this, the P is silent. Okay, so we call this a ptyalin and a pancreatic alpha amylase. What is ptyalin? It is act as a linear alpha 1,4 glycosidic bond to break the large insoluble starch molecule into soluble amylodextrin, erythrodextrin, and archaeodextrin, producing successfully smaller starches. Pancreatic alpha amylase. It randomly cleaves the alpha 1,4 glycosidic linkage of amylose to yield dextrin, maltose, and maltotriose. The alpha amylase are calcium metalloenzyme function only in the presence of calcium. These are the diagram. This is the amylose and it is the 1,4 glycosidic linkage. Okay, it is a straight chain and this is a branch chain of amylopectin. It contains not only 1,4 linkage but it also contains 1,4,1,6 along with 1,4,1,1,6. Okay, glycosidic linkage. This is the branch point of alpha 1,6 glycosidic bond. And remember one point over here. It is not only the single enzyme, there are many enzymes which act on it and cleave the polysaccharide chain into the smaller component so that our body can easily absorb them. Okay, I'll let you know what are the other enzymes. Okay, our digestive system utilizes protease to break them down into the smaller component. One of them is salivary alpha amylase and it is going to cleave alpha 1,4 glycosidic linkage in the mouth and break them down into, okay, I will write and you kindly also note it down. The first is salivary alpha amylase. It broke down the polysaccharide chain into the mouth, that is the alpha 1,4 glycosidic linkage into smaller component of what? Into smaller component of polysaccharide and oligosaccharide. Polysaccharide and oligosaccharide. Oligosaccharide. Okay. The next is pancreatic alpha amylase. Pancreatic alpha amylase. What it is function? The pancreatic alpha amylase release more powerful alpha amylase into small intestine and break them down into disaccharides and trisaccharide. Disaccharide. Can you name one disaccharide? Do you know? Okay, if you don't know, its name is maltose. Okay, and trisaccharide. Come on, tell me one name of trisaccharide. Its name is simple, is maltotriose. Okay. Then the next one is maltase. Maltase. Its name is so simple. What its function is? These enzymes break down these maltose into its glucose constituent. So their basic function is break down of these maltose. Okay, maltose into simple glucose constituent into glucose. Okay. Before I tell you more, please look at down this diagram. This is a diagram of a small intestine. It contains the villi. I am mainly concerned about the brush border. Can you see the brush border of microvilli? This pink color, this whole structure. This is the brush border. So when our body consumes these polysaccharide and the breakdown occurs, these small intestine at the brush border absorb it, and all these enzymes are present over here. Okay. The next enzyme is alpha glucosidase. Alpha glucosidase. These are also present at the brush border and their function is digest maltotriose into what? Simple. Maltotriose into simple what? Do you know? Glucose. Easy. Then the next one is alpha dextrinose. Okay. Can you tell me what is the function of alpha dextrinose? In this diagram, see, I have told you the starch contain two linkage, right? In amylopectin, it contains two linkage. The one is alpha 1,4 glycosidic bond and the second one is alpha 1,6 glycosidic linkage. The alpha amylase can only break the alpha 1,4 glycosidic bond, but it is unable to break this bond, alpha 1,6 glycosidic bond. So the enzyme which is used to break or cut this bond or cleave this bond, the name of this enzyme is alpha dextrinose. So the function of alpha dextrinose is it break down alpha 1,6 glycosidic linkage, or you can say alpha 1,6 glycosidic bond. This enzyme is sucrase. Its function is break down sucrose into glucose and what? Do you know it is a fructose or galactose? Yeah, you are right, it is fructose. Next one is lactase. What its function? Its function is to break down lactose. From where we can obtain the lactose? From the milk, right? It's easy. Into glucose plus glucose and galactose. Okay, these are the basic list of the enzymes which help in our digestion and through which we can ultimately consume the glucose. We can uptake the glucose. These are the enzymes which help to cleave or breaking down the polysaccharide chain into the smaller component by which the glucose can easily be uptaken by the microvilli which are present at the brush border of a small intestine, through which the glucose can easily exported or into a lymphatic system. Now you can easily understand what is the amylopectin, what are the amylose, and what are the enzymes that are required to break down the polysaccharide into smaller component. I hope there will be no doubt. If you really have any doubt, please let me know in the comment section below. Thank you for watching the video.